Okay, so I thought I'd give you an introduction to our Kgofa project. If you search this online, currently our funding is under the long title of Revolutionising Livestock Tick and Tick-Borne Pathogen Control. And it's a collaboration between many institutes. So we've got quite a big team. So there's myself and Professor Al Darby at the University of Liverpool. I'm a parasitologist, and Al Darby is the director for the Centre of Genomic Research at Liverpool. And then we also have two postdocs, Maddie and Adam, who are not here today, our lab technician, Alice, who does all of the hard work, and then, of course, Lingasisi at Ori, Casper Nyamakondiwa at Bust, and they've both been supported by Precious and Virgil in the field. And then we have Lewis Neves, who is a tick expert at the University of Pretoria. So to give some global context to this, there are more than 850 species of ticks identified globally, and about 150 of those are found in Africa. And these are incredibly diverse ticks. So see, these are some of the pictures that Alice has taken of the ticks that were collected in Botswana. And of those 150 ticks, 120 of those have been reported on ruminant livestock. So a very diverse range of ticks infecting our livestock or your livestock. And what I'm most interested at the moment are the Amblyomma species, so Amblyomma hebræum and Amblyomma variegatum here. Other than them being very beautiful, if you like that sort of thing, they are, of course, responsible for heart water and associated with the dermatophilosis. And in terms of the economic impacts, East Coast fever costs an estimated $168 million per year in just 11 African countries, and is estimated to kill around 1.1 million cattle per year. Obviously that's an underestimate because it's only an estimate for 11 countries. And this is a nice infographic from ILRI, who are currently developing diagnostic methods for East Coast fever, which is caused by Tullia parva. And then heart water, which of course you have here, is estimated to cost South Africa $72.5 million per year, and Zimbabwe $5.6 million per year. And what these figures don't really tell you is the human cost to this and the animal welfare cost, which Lingasisi touched upon, the livelihoods of the farmers, especially the rural communities that are affected by these diseases. So what about dermatophilosis? So Lingasisi already covered this, so I won't go into too much detail, but there are many factors involved in the epidemiology of dermatophilosis. The main risk factor being warm, wet weather, but we also have different breed susceptibilities. So some studies have found that Friesians are more susceptible to dermatophilosis. Poor nutrition, stress, and anything impacting the immunity can, of course, impact susceptibility. Mechanical trauma to the skin, and there's also potential for co-infection. So there are a few case studies published in camels and cattle elsewhere where there have been co-infections with bacteria, fungi and viruses as well, which makes the management of this disease very difficult. So ticks are not required for dermatophilosis to be present, but they do facilitate disease. So they cause mechanical trauma to the skin, and also they impact the immunity of the cattle. So the mechanical trauma is through the feeding action of the ticks as they penetrate the skin. Amblyomma ticks, the Bont ticks, they've got really long mouth parts compared with many other species. So they cause a lot more trauma than other species. But only Amblyomma variegatum has been found to be associated with dermatophilosis, and not Amblyomma hebræum, which you also have here around, for example, Palape and Saroa. So a study in Zimbabwe found that herds that were infested with Amblyomma variegatum ticks were 7 times more likely to show clinical signs of dermatophilosis, but this was not found in herds that were infested with Amblyomma variegatum. So there's something else going on here. And this is related to some observations that we sometimes get questions about why, if the disease is associated with ticks, do you also see lesions on other parts of the body that are not infested with ticks? And that can be because of any one of those factors on the previous slide, but it can also be because amblyoma variegatum has now been shown in several controlled laboratory and artificial infection studies to immunosuppress the cattle. And so you get this systemic immunosuppression, which makes them more susceptible to infection in the first place, and then more susceptible to severe disease as well. I've produced a fact sheet on this that is just about the tick risk factor, not any of the other tick risk factors, but just because we do get this, these questions quite a lot. So I've just done a very, very brief literature review on this, and you can access that on our Zenodo website or using the QR code. I've also printed some here today. So how this project came out, that's the importance of ticks, but how this project came about was I first visited Botswana and sort of reconnected with Lingasisi after doing our PhD in the same group in 2017, and I was just assisting with another PhD student's project on nematodes in goats and nematode control in goats. And then I saw ticks and I became interested in this, and we came back the next year and hosted with BNVL a workshop on food security, parasites and tick-borne disease. And what everybody wanted to discuss at that meeting was the tick-borne disease and biosecurity issues, and that was in 2018. And so there are two amblyoma ticks that are present in Botswana. Amblyoma hebræum on the right, and you can see the dark green is where we expect amblyoma hebræum to be found, and amblyoma variegatum on the left. And that should only be found around, say, the Kasane area, with the border between Zambia, Botswana, and Zimbabwe. But that wasn't what the vets were seeing at the time. But they didn't feel confident to identify or tell the difference between these two ticks. So you asked if we could come back and do some tick identification training, which we did the following year. One workshop in Maun and one in Gaborone. And we asked the vets to bring their own ticks from their own district to identify and to practice. And one vet brought us an amblyoma variegatum, which we were really excited about, from Betsa. And amblyoma variegatum shouldn't be in that area, according to our existing maps. So clearly we needed updated distribution maps, certainly so that vets in the different districts had some idea of which ticks to expect, and therefore which tick-borne pathogens to expect, and then that would help you with your diagnosis. Has that gone to the next slide? No. And then in 2019, Virgil and Casper Nyamukundiwa, we worked together to do a pilot survey of ticks throughout Botswana. So they went to 11 villages, 10 kraals in each village, and Virgil's going to present the results of that after our break. And then that brings us to this project. So finally, 4 years later, in 2023, we managed to get some extended funding for a 3-year project. We're now 2 years into that. We've got a year left. And so we're just going to tell you some interim findings from this project so that you don't have to wait years for publications. Scientific publications quite slow. And so that we can get some input from you so that you can shape what we do for the rest of the project, if possible. So the project is looking at 3 things, broad aims, and we're really showcasing Botswana as a case study of what can be done when we work together as scientists with the veterinary services and with the policymakers. So first of all, we want to better understand the microbial communities in and on ticks, and that includes tick-borne pathogens. So the first of that is nationwide tick surveys, and I really just want to congratulate all the field teams who did this. As far as we're aware, this is the first truly randomized, spatially unbiased tick survey nationwide in any country in the world. So it's really a world first as far as we're aware. And then they became aware of why people don't normally do this because it was very challenging, very exciting, but very challenging. And there's stories of lions chasing the vehicles and multiple breakdowns and having to borrow a donkey and ride it 20 kilometers to get help and things like that. So that's why people don't do these things, but they've really persevered, and I congratulate them on that and thank you very much for that. Yeah. So Alice is going to present the results from that. She's been identifying all the ticks in our lab at Liverpool. And then the next step, which is next week, we're going to send off our first batch for sequencing, and we're going to sequence the entire bacterial and protozoal community within each tick individually, and that includes the pathogens. So we should be able to identify all of the pathogenic bacteria, all of the pathogenic protozoa, assuming the sequencing works, within each tick, and then give you those results as well. Next, we're going to model, or we've already begun modeling tick-borne tick populations, so we can predict the seasonal variability of ticks, so you know exactly when to target tick control, and tick-borne pathogen transmission. So the beginning of this, which I'll present later, is species distribution models. This is a machine learning approach. It's a type of AI, and it allows us to look at the climate suitability throughout Botswana. We've actually done it on a continental scale. And then we use that to underpin our mathematical models of tick environment, pathogen, and host interaction. So a very complex system. That's the one that's taking time, but that's something that we can get input from you on. And the final stage of this project is to develop evidence-based recommendations for tick-borne pathogen control. Now, the FAO have produced guidelines for global guidelines for tick control, and they released that just a few weeks ago. So it's really good timing with this meeting, and we've got Letitia, who produced those guidelines, presenting those to us later. But they really need to be tailored for Botswana and for what's possible in Botswana and for your needs. So we need to incorporate our knowledge from this project and from other studies on tick and environment and pathogen and host interactions. And we'll use our models to virtually compare and test different veterinary interventions and different policy strategies to control ticks and tick-borne pathogens. So that's where we could really do with input from you. Which policies do you want us to compare? Which interventions do you want us to compare? And then we make sure that it's useful for all of you in the end. So we'll run through that the rest of the day. I'll leave it there and then pass on to the BNVL. But there are many people involved in this project other than the people that I mentioned on the first slide. So many advisors, and of course the DVS and the DWMP, who have been instrumental in facilitating our research here over the past couple of years. So thank you very much.